My name is Richard Sokolov. I'm a director at IDE. We're a product development services firm. We've been doing the development of the MicroRapid for Atomo Diagnostics, and I've been leading the team on that technical development of the product.
My name's John Kelly. I'm the founder and Chief Executive Officer of Atomo Diagnostics. I've been involved from the start with the idea, with the development of the concept, and with working with IDE to make sure that the solution that I had envisaged could be actually delivered in reality. I have a medical device background and just felt that it didn't need to be this hard, that there was a better way to access blood for diagnostic testing. And that really started the adventure from there. Our device is a platform device that can be used to test for a wide variety of diseases. Currently there are in the market commercial lateral flow test strips to test from anything from HIV through to cardiac markers. Our device has been designed to accommodate those strips and provide them to the clinician in a much more user-friendly format, and by doing that improving the usability and hence the repeatability of the procedure. So this is the MicroRapid HIV device. Richard is starting by sterilizing his finger with an alcohol wipe, which is standard for any blood collection procedure. He is now removing the sterility tab, which stops inadvertent activation of the lancet. The lancet is now activated by push contact, simply by pushing the device directly onto your finger. That's now punctured and retracted back into the device and has generated a blood sample. And Richard's now in the process of generating a blood sample by gently milking the finger. The finger's now generating a blood sample, and that blood, when delivered to the device, automatically pulls down using capillary action, and the blood collection system collects exactly the right amount of blood required for that particular test. In this case, 10 microliters. Once the device is full, no further blood collection is needed, and the device is now ready for delivery of the blood automatically onto the device. You simply rotate the BCU, it clicks onto the device. Capillary action in the strip is greater than in the tube, so it automatically drains within about 10 seconds onto the device. The blood's now in the strip. To get a result, a buffer solution is dropped into the inlet port, and this then chases the blood down into the test zone, and a result will be delivered within 10 minutes.
The main functional issue or technical issue that we have to overcome is the actual usability of the device and making it intuitive to be able to actually use the device easily. I think previous versions of the same type of test, the kits that are available in the market, make it very difficult for you to use it repeatedly and accurately. So a lot of the prototypes you'll see in the display show the evolution of our thinking, and even the sketches will start to show you what we were thinking in the beginning. So you'll sort of see some of the very early mock-ups or prototypes that we did didn't have everything integrated, or had too many things integrated at times. And so basically by using usability trials and working with people in the field and trying the prototypes, we're able to nut out and sort of eliminate what was excessive and integrate what was necessary to give this sort of seamless, more user-friendly, intuitive device that you sort of see in the final embodiment of the product. You'll see some very rudimentary foam prototypes, which are just, people would think is very useless, but actually quite revealing as to how to use the device. But once we progressed and knocked out some of the easy issues to identify with ourselves and with people in the team, we went to people in the field, so nurses, clinicians, and we also just did some family and friends as well. People who didn't know anything about the device, who hadn't had the experience, and some people who had had experience with, for example, doing diabetic tests on themselves, to give us a sort of a range of perspectives on how this device may be used and what are the problems with what we had developed to date and what to do to actually improve upon it. For those people who have not used the device before, it is much more intuitive than what the current products are out there in the market, and further development is going to actually improve on that. So the first embodiments that you see in the display are for clinical screenings, so it's geared towards the clinician. And future devices may be geared more towards lay people and improved even further.
There are a lot of lateral flow test kits available. The most common one would be a pregnancy test. But a lot of the assays on the test strips require blood, and they require a developer buffer to chase the blood down to the results window. In point of care settings, although the test cassettes themselves are very accurate when done properly, the variability in using them in the field creates a lot of variability in the quality of the results, and performance in the field is typically with a kit not as good as in the lab. And the reason for that is you have to use a manual pipette, you have to use a lancet to generate the blood, so there's a lot of ancillary equipment that is needed to support the test, and we decided that those ancillary equipment resulted in a lot of complexity and a lot of handling and a lot of sources of error, and our rationale from the start was to try and integrate all of those kit features down into a single device and take the need to handle multiple items out of the procedure. The concept was from the start to develop a product that would be easier for a clinician to use, but would also offer the ability for a self-test application. There is a transition now away from centralized hospital services out into the community, and this type of easy-to-use test supports not just health workers in the field, but also potentially home-based testing. Our strategy is, once we have the HIV product launched, to look at other applications that we can put directly onto our device. Malaria is one, and we're also working with a couple of companies in Europe around applications for things like celiac detection and allergy detection.
The materials that the device is made from is actually sort of your common plastics, injection molded plastics. The reason for that was that basically we had to keep the cost of the product very low to be able to be effective in sort of, let's say, not a first world or a third world type of scenario where people are screening large populations. The cost is always a major impact. So to be able to be using that in that sort of context, we had to make it the cost as low as possible, and therefore we were forced into using sort of more general type of polymers. And we've managed to achieve all our technical goals with that. Some have been very, very difficult to achieve, which you might not see through just looking at the device, but we were able to accomplish that and keep it in the realms of commercially viable product. Not all the components are just polymers. If we look at the puncture device, that's a stainless steel. It's a medical grade stainless steel. And basically it's gamma irradiated to sterilize it. So basically it's, once it punctures you're protected from any sort of bacterial infections. It also retracts into the device, so it prevents any sort of cross-contamination or potential sharps infection to the actual clinician applying the test. And also the test strip itself, there's nitrocellulose membrane inside there that basically wicks the blood and diluent mix through the actual test strip, and then it binds with different elements inside the test strip, which are actually dried onto the test strip, freeze-dried onto the test strip. And they also have antibodies and different detector agents, which then take the sample through and gives you the result in terms of lines, whether you're positive or negative. So I guess that was a technology that was already there, that we're basically integrating into our device. The portion of the device which actually manages the blood, delivers it, and then also embodies the integration of all the elements, that was where our main technical challenge was. And then obviously not to disturb the actual process of the test being performed on the actual strip. So there are a number of key mechanical and technical innovations there to make sure that maintains its integrity.